nightmare.
Activate.
Hello guys, welcome back, and now we got question number 3 from the 2019 biology paper 1 higher exam question. Okay, so question number 3.1, complete the word equation for photosynthesis. Now notice that it's asking you to write down the word equation. It's not asking you for symbols. However, if you write down the symbols, you might still get the marks, but you always got to do what the question is actually asking you. Okay, now we know that in order for photosynthesis to happen, plants are going to need water, and they're also going to need carbon dioxide. Okay, and now one of the products is going to be oxygen and the other product is going to be glucose. So you have it, the first two easy marks. You get 1 mark for getting this part correct and then 1 mark for getting this part correct. Okay, let's go to the next section. Says a student investigated photosynthesis using pondweed. Figure 3 shows the apparatus that the student used. Now when we say apparatus, we're looking about the whole setup of the equipment. Okay, then it says you have your light bulb at 60 watts, and then you have your water, your funnel, your pondweed, and then you have the little bubbles of oxygen going upwards, and as the oxygen bubbles go up, they push the water down, and with this measuring cylinder you can measure how much gas is actually being produced. Okay, because the water level is being pushed down, right? So it says there, this is the method used. Set up the apparatus as shown in figure 3, switch on the lamp. After 20 minutes, record the volume of oxygen collected in the measuring cylinder. Repeat steps 1 to 3 using bulbs of different power output. Okay, so this gives us question 3.2. What was the independent variable in the investigation? Now let's remember that the control variable is what you keep the same, the independent variable is what you change, and the dependent variable is what you're measuring. Okay, so in this specific case, the thing that we're changing is the power output of the light bulb, because that is the thing that we're changing. And notice it says tick 1 box. Okay, then we go to question 3.3. Suggest 2 ways the method could be improved so that the results be more valid. Now there's a number of things that you can mention over here. One of them is that you can repeat the experiment so you can calculate the mean, and that way you can eliminate any anomalies. You can control the temperature of the water, control the concentration of carbon dioxide in the water, control the distance of the light bulb from the pondweed, control the mass, length, species, age of the pondweed as well, and give pondweed time to equilibrate. Okay, that means that you're just gonna let it settle in the water for a couple minutes so it gets adjusted to the temperature and everything else, and then you can start your experiment. Okay, now if you want to write 2 of the simplest ones, you want to go for the control of carbon dioxide and control the concentration of carbon dioxide, and then you want to also go for the repeat and calculate to get a mean. Okay, all right, those are the 2 ones that I'm going to go for. Let's go for question 3.4. Question 3.4. The table 3 shows the student's results. Power output of light bulb in watts. You got 60 watt, 100 watts, 150, 200, 250 watts. These are the all different light bulbs that he used. Okay, so in total he used 5 different light bulbs. Then you have the volume of oxygen collected in 20 minutes in centimeter cubes. So that one in 20 minutes made that much oxygen, then the 100 that much, 150 that much, 1.2, and then after the 200 and 250 they both had the same amount. Then we got the rate of photosynthesis in centimeter cubes per hour. You got 1.5, 2.4, then you have an X, 3.6, and then 3.6. Calculate the value of X in table 3. Okay, so how are we going to do this? Now, first of all, we're going to look at the difference between 1.5 and 2.4. What's the difference there? The difference is going to be 0.9, okay? And then if we add that plus the second value, this has increased by 0.9. So let's try to increase the second result by 0.9 as well, because it will probably be the next option as it's increasing by 50. So this is increasing by 10. By 40, and this one is increasing by 50. So let's just have a close estimate of it, and if you do 0.9 plus 2.4, you're gonna get 3.3. So that will be your answer in there. So we go to 3.3. There you have it. Let's go to question number 5. All right, so then we got question 3.5. Complete figure 4. You should label the x-axis, use the suitable scales, plot the data from table 3 and your answer for question 3.4, and then draw the line of best fit. Okay, so let's have a look. So what we got here, first of all, is your y-axis, and this is what you are going to be measuring. Okay, your control variable is what you keep the same, and your independent variable is what you're going to change. Okay, in this case, what we're going to be changing is the power output of the light bulbs. So that is what we're going to have over here at the bottom in the x-axis. Okay. So there we have it. Now we gotta make sure that our scaling is well done so we are not making any mistakes. A lot of people, what they tend to do is that they will just put down the numbers straight how they are in a table, and they will always end up with a straight line with the positive correlation. All right, and that is always going to be wrong, because that's not always going to be the case. Okay, if I were there to be doing that, then if I end up with a straight line, it will indicate that everything is directly proportional when it's not. So avoid doing that. You gotta make sure you get the right spacing in the right scaling in your graph. Okay, now if we look at the big squares over here like that, you got those big squares, right? If we count each one of those as 50, let's see how far we can get. Remember that the biggest number that we need to write down is 250. That's the biggest number in the table, so there's no point if you're going all the way to 1,000, because that's just going to be wasted space. Right, so we got 50, 100, 150, 200, 250. That seems sensible. So let's have a look. Remember that our first number is actually 60. Our first marker is 60. So where do we put 60 over here? So let's have a look. Now, if we count every single one of those spots, you go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. If you count as one each, it's going to be a bit too long. Okay, so we can count them as twos. And if you count them as twos, and we think that, let's start from 0. If that was 0 there, and we count them as twos, you're going to say 10, 20, 30, 40, 50, 60, 70, 80, 90, 100. Okay, so that's a pattern that does follow. Okay, if you done ones, then you wouldn't match the rest of the numbers. So if we go there and let's mark it off and go for 10, 20, 40, so 10, 40, 60, 80, and then we got the 50 there. So again, I just made that confusing. So if we count them out, we would say 10, 20, 30, 40, 50. And then we're going to go to 60, 70, 80, 90, 100. All right, so let's use those scales in order to get this. Now, obviously, that one there, if we follow across there, is going to be your 60 mark. And now all we have to do is plot the power output of the light bulbs against the rate of photosynthesis. Okay, and you've got to make sure we get this correct. I'm going to use a ruler to make sure that I follow the lines appropriately, because sometimes you can get confused as you're looking at too many squares, and then you just get confused of where the dot should go. But using a ruler is going to help you get that more on point. Okay, so the first value, when it's 60, the rate of photosynthesis is going to be 1.5. So again, if we go these ones, that will be 0.5, 1, 1.5, and then you got 2.5, and then 3.5. Just gonna mark those in advance. So at 60 is 1.5. So let's have a look over here. If I trace that 60 all the way to 1.5, which is that one there. Okay. Now, dotted line, and then I'm gonna do oh, look at that, I made a mistake. That's 250. So this is why it's very important to do this, just in case you get it confused, and then you still know where that line is gonna be. Okay, so when it was 60, we look at 1.5, and then that is where our first dot should be. Then at 100, we're looking at 2.4. So 2.4 will be 2.1, 2.2, 2.3, 2.4. It will be there. Okay, so let's go 100, which is in this line here. Somewhere there. We don't even have to do it exactly where it is. because then we're gonna find out once we draw the other line from 2.4 and that's that. Okay, so we're gonna do the same for the other ones, and we're gonna check out how it ends up as a result. Okay, and there you have it, guys. Those are your dots plotted correctly. And now the other thing that you want to keep in mind is that it says in here, draw a line of best fit. And a lot of the time people get that confused and they start doing straight lines of best fit, like putting as many dots as possible through the line, but that's not what it means. Now in this case is blatantly a curve of best fit, because the line is starting and is curving until nothing else is happening. Okay, so that is what you want to do every time you see in science, draw a line of best fit. Unless you see a dead straight line or straightish line, you're gonna have to do a curve of best fit. Okay, otherwise you're gonna get that wrong. And it's not a dot to dot line. Okay, it's a smooth curve, as smooth as possible. And there you have it. Okay, now it says question 3.6, determine the expected rate of photosynthesis with a light bulb of a power of 75 watts. Use figure 4. Okay, so 75 watts, what do we have to do? We need to look for 75 here in the graph and then try to fit it into the pattern. Okay, so if that one is 60 and this one here is 70, 75 will be this one here. Okay, that will be your 75. Okay, so once again we trace the line from 75 there you go. Okay, so according to our graph, from 75, if you follow up until the line, until the curve of best fit, you stop it there, you go across, and then we end up with 1.9. Okay, there you go, easy peasy, easy marks. Okay, right, so let's go to the next question. Okay, question 3.7, which graph shows the effect of temperature on the rate of photosynthesis? Okay, now we know that when we increase the temperature, the photosynthesis is also going to increase, but it will get to a point where it's going to be too hot, and instead of continuing to increase or staying the same, the temperature, the rate of photosynthesis is actually going to go down because the enzymes are being denatured because it's too hot. So definitely it's not going to be this one, right? That's just a straight line. This one makes no sense as well. It says the photosynthesis is going to be decreasing as you increase the temperature. That is not no as well. We got this one here that makes no sense. It says that the temperature, as you increase the temperature, the photosynthesis is going to go down, and then at some point, as it reaches the highest temperature, then the photosynthesis is actually going to go up. Okay, so this is the main one. This is what we're looking at in this question. It will be that one right there. That is your answer. So as temperature increases, the rate of photosynthesis also increases, because if we had numbers there, it will be increasing, and it gets to a point where the temperature is too hot, the enzyme is going to denature, and then the rate of photosynthesis is going to go back down. Okay, so that is your answer right there. Okay, so that was question 3 from the 2019 Biology Higher Paper 1. Let's move on to question number 4, and I'll see you in the next video.
It was a nightmare. Activate.